All right. I'm now trying to get the YouTube live stream started, and then we'll kind of start shortly. All right. I think we're going to go ahead and get started. And so with that, I welcome everyone and thank you for joining us in our next webinar in our webinar Plantae webinar series. My name is Jason Padilla and I'm your technical host for today's webinar. Next slide. This webinar series is brought to you by the American Society of Plant Biology through Plantae. Plantae is our open online community for plant scientists and plant scientists. I would like to give a special thank you to all our ASPB members who are attending today's webinar. Your ASPB membership dues help support making this webinar possible. For any of you who have not yet joined ASPB, you can join today with a discount code and receive a 10% discount on your registration. ASPB members get early access to this webinar, and you can learn more about all the work that ASPB does by going to aspb.org. Next slide. This webinar is recorded, and we've had so many amazing webinars in the past. So if you're looking for great plant science content or plant science related adjacent content, don't look no further. So just check out the Plantae YouTube channel and start browsing some of our content for our past webinars. Next slide. Before I get started, I'd like to go over a few housekeeping items to make sure that you get the most out of your time when attending today's webinar. If you are experiencing technical difficulties, please let us know about those in the chat box, or you can email me at jpadilla@aspb.org. If you have questions for our guest speakers, please add them to the Zoom Q&A section, which you'll see on your screen. Our moderator will be sharing them with our speakers, and we'll try to get those answers for you all. If you are having trouble connecting or need to leave early, as I mentioned, this webinar is recorded and will be made available later on our YouTube channel. So be sure to follow the Plantae YouTube channel so that you know when content are up. And so without further ado, I will now turn it over to our Plantae Fellows who will be moderating today's session. So take it away, Plantae Fellows.
Hello and welcome to this webinar. My name is Ileana Tosolini and I'm going to be your moderator today together with my Plantae fellow colleagues, Bilo Bernard and Jonathan Villas-Boas. And this webinar will focus on the landscape of plant science research across different parts of the world through talks with these notable researchers who have developed their scientific career in diverse regions, Dr. Federico Ariel, Dr. Sonia Negrao, and Dr. Felipe Kliemann. Our aim is to uncover the realities faced by scientists in different geographical contexts and how these regions can shape the nature of research. First, I'm going to give a quick overview of the topic. Well, doing science in different parts of the world offers unique experiences and challenges. And as we know, opportunities, research topics, and funding are not the same and vary depending on the location in the world. And it's not just about the science, it's about the entire ecosystem that surrounds it. And for scientists born in countries where research and innovation are highly supported, the scientific journey may appear more straightforward. Of course, not always, but for those hailing from regions where science takes a backseat in national priorities, their career paths often have more obstacles and challenges. At different moments of their career, many researchers may have to decide whether to move to another country or institution to start a new position. And here some questions come up. For instance, is moving a good idea? When and where to move? And in the past years, research activities have become increasingly globalized. This can be observed via two phenomena: international scientific collaboration and international mobility of researchers. There are many reasons for scientific mobility, like access to better resources and funding opportunity, career advancement, expanding professional networks, increasing visibility within the global scientific community, cultural exchange, and more. And for many years, the concept of brain drain has been the prevalent interpretation of this phenomenon. And from this perspective, the movement of scientists is attributed to economic reasons and is associated with a transfer of human capital from sending or losing countries to receiving or winning countries. However, recently this term has been replaced by the concept of brain circulation. This means that scientists move around the world, but they also come back to their home countries, bringing new ideas and skills. So international mobility benefits all parties. For example, in this work, they conclude that maintaining home connections, such as engaging in teaching activities in the home country, maintaining collaborations with academia or industry, or having return policies based on economic incentives, increases both the probability of return and scientific productivity in the host country. Although under certain circumstances, several factors can be an obstacle to the career progression of mobile and foreign scientists, like language, the local definition of research quality, travel visa restrictions, cultural differences, family concerns, and also homophily, which implies that people have a preference to collaborate with those who are perceived to be similar. So addressing these obstacles requires measures in order to promote inclusivity, diversity, and unbiased opportunities. So to further expand this webinar's topic, we ran a survey and 40 plant scientists from around the world participated. Their areas of research were diverse, but the majority are focused on plant responses to stress, genetics and genomics, and plant development. And despite being a small sample, we had a relatively diverse participation from scientists born in several regions, which are marked here with circles proportional to the number of researchers surveyed. And the majority of them, the 87%, have conducted research in a different country, including short stays. And of those who went abroad to conduct research, 51% ultimately returned to their home country, while the remaining 49% are still working abroad. And on the map, we can see arrows indicating their current location. And this allows us to observe the trends of scientific mobility among the respondents. We can see like a trend towards regional mobility here, and especially in Europe and South America. But however, we don't know if they have migrated more than once and to other locations in previous years. There are several studies that have investigated scientific mobility. For example, in this one, the authors found that nowadays, internationally mobile scientists move more frequently to other countries, but over shorter distances, what they call regional moves. And also they saw that over time, the migration flows to and from Europe and Asia have grown, while the flows to and from the US have decreased. This work also indicated a similar trend, and they also observed that while female researchers continue to be underrepresented among internationally mobile researchers and migrated over shorter distances, this gender gap has been narrowing faster in the past years. And this work revealed notable geographical bias in plant science research that is associated with national affluence. And they saw, for example, that most countries prefer to collaborate with researchers in Europe, Northern America, or China, and were less likely to collaborate with authors in Latin America, Africa, or West Asia. And in our survey, we also asked if they have ever thought about doing research abroad, and only 5% answered no to this question. And we also asked about the reasons why they left or would leave to conduct research in other regions. And the most voted options were for better research opportunities and networking, career advancement, access to advanced facilities, that is labs, equipment, and resources, and cultural exchange too. Then we asked researchers to summarize the pros and cons of doing research abroad in 3 words. And we can see that better facilities, networking, more opportunities, professional growth were among the main advantages of doing research in other regions. And the most mentioned disadvantages were related to being away from family, homesickness, language barriers, and cultural differences. So to conclude the survey, we asked if a person's geographical location might influence the nature of research and why. And only 2 people answered no. Well, availability of funding and facilities were among the most mentioned reasons. This is connected to research efforts that often focus on regions' challenges, interests, or needs when funding is limited. Also, the nature of research might be related to a local question or phenomenon bound to the location. And we can quickly see in this figure that there are national differences in focal organism choice. And this study of Marx and collaborators found us that most papers focused on major crops and model species, while the remaining biodiversity accounted for only a fraction of publications. And for example, a scientist from Peru said to us that they received poor financial support despite being in a country with the highest levels of biodiversity in the world. Another researcher from India mentioned that they hardly get better funds for plant ecology compared to animal-focused wildlife ecology and also biotechnology-based plant biology. And finally, I want to remark this answer we received that says our cultural backgrounds shaped by geographical location, but greatly influence the way we think and see things. And that's why international collaborations are really important for delivering impactful science. So now we are going to hear our notable guests and discuss about the challenges of conducting research in different parts of the globe. And it's an honor for me to introduce our first speaker, Dr. Federico Ariel, group leader at Instituto de Agrobiotecnología del Litoral in Santa Fe, Argentina. Well, he just moved to the IFIBINE Institute yesterday, which is located in Buenos Aires. His research is mainly focused on studying the evolution of long non-coding RNAs and their role in the dynamic 3D organization of the genome. Dr. Ariel also co-founded Apollo Biotech, which is dedicated to the development of RNA-based technologies for protecting crops from pathogens and for enhancing the resilience of plants to climate change in a sustainable manner. Dr. Ariel has conducted research in France and in the US, and he has won several national and international awards, including the recent UNESCO Alpha Prize last year. So let's welcome Federico, and thank you very much for participating in this webinar.
Thank you very much, Ileana, and the organizers for this invitation, and ASPB in general. I'm very impressed by the results you have shared. Thank you for that research you've done, because it's very informative and it gives us a very important idea of what is going on around the globe and plant sciences. So thank you very much. It was very, very interesting. So as Ileana mentioned, I'm from Argentina. I did my studies in a city far from Buenos Aires, that is the capital city, where like in many other countries, most of the research is going on in the capital city. I was born in another city and I studied in another city, so I did I obtained my degree at university and then in biotechnology, and did my PhD in plant molecular biology in that city. Mainly focused on legumes and the symbiotic interaction with bacteria. And then I obtained an EMBO long-term fellowship to move to France were to join Martin Crespi's lab in Paris to work in the non-coding genome of plants. By then, with all these new technologies of next generation sequencing, we learned that although most of the genome doesn't code for proteins, it's being actively transcribed into RNA, and we were like looking to this non-coding genome and try to figure out what these long non-coding RNAs or small non-coding RNAs were doing, what were these roles. So we mainly contributed to the understanding of these molecules in epigenetics and alternative splicing. I stayed in France for 6 years. During that period, there was an active network that was conforming of Argentinian researchers around the globe, forming like groups in different countries. So I joined this group in France. So they were like postdocs or senior scientists that were already settled down in France, that were gathering and trying to promote collaborations between France and Argentina. And then in 2016, it's been already 8 years now, I came back to Argentina. And I came back to Argentina within in the context of a program for helping young scientists to come back to Argentina. So it was easier to obtain a permanent position in the research council, that is CONICET, or also to have a position at university, but also the moving from one country to another one was, we had money for doing that, and we had special grants for the installation grants. So this was very effective. So when I came back to Argentina, I joined this program, and I'm still part of the advisory committee of this program in Argentina. And over 1,500 people came back to Argentina within this program. So very, very effective. And I agree with what Ileana said before, that there are many ways to contribute to our home countries, to the scientific system of our home countries. One can be to participate actively in our countries, like I'm doing right now, but also we keep collaboration with 23 other countries where there are these groups of Argentinians that participate in this network, and we promote a lot of networking around the globe. We help a lot with infrastructure, fellowships, and we share a lot of things. So that was very, very important for me. It is still very important. And there are some things that I realized now that I'm working in my home country that perhaps I was lucky enough, but I found a few aspects of my career that were very helpful, and I would like to share this with you right now. First of all, that when I chose my postdoc lab in France, I did it only because I knew Martin, that was an excellent scientist and was a leader in long non-coding RNAs in plants. But also, now I realized that it was very helpful to come back to Argentina and to keep on working with Martin, and that he was very, very nice towards me, because he allowed me to continue with my research lines in Argentina. So conducting my own research, leading the lines, and that helped me to avoid having a gap in the scientific production. I was not moving to Argentina to start a new line. I was continuing with my line in collaboration with Martin. And that was very important. And I know colleagues who came back to their own countries or went to another country after their postdoc, but they had to leave behind all they had done and start new research. And that is really important. So if I had to choose now again a postdoc lab for my, of course, I won't do it, I would take that into account. What are people who went through these labs are doing right now? Do they continue to work with these people, with their former directors? Do they collaborate with them? Are they expanding their networking, or they had to leave the networking behind to start a brand new project without any collaborations? And I say this because, as Ileana mentioned before, there is a big problem in the global south for scientific funding. Right now, Argentina is going through a very difficult situation. There are a lot of cuts on budgets, and being part of an international network is very important to keep on doing research, to have access to platforms, to being able to participate in international conferences, and also because we can receive to host in our own lab in Argentina students from other countries. Now we have a postdoc who is from Colombia, we have a PhD student from France, but we have also short-term visitors from Italy, from Germany, from France. And that is very, very nice because diversity matters. In all terms. So I think that all this is very important. Right now, we are part of an international associated lab founded by France and Argentina. And all this, I think it's helping a lot to keep on working with different labs around the globe. At the same time, I still observe that there are problems that I would love to fix within the Argentinian community. One of them is that it is more, it's easier to find funding to collaborate with Europe or the US rather than with Brazil, Uruguay, or Chile. And that is very weird because we are just next door, but there are not programs or very few programs that we can apply to conduct research in collaboration with our neighbors. Although I love to collaborate, especially with France, we have this international associated lab, and I want to keep on doing that. I would love to expand our networking in a regional matter, in a regional aspect, because sometimes the problems we have to face are different in a region from that is important in Europe. No, for example, Brazil, Argentina, countries with a lot of where agriculture is very, very important, and perhaps the problems we need to address are different from those in North America or in Europe. So I think that that's something I would love to improve. I try hard to collaborate with the neighbors, and I really like it. And I think we need more funding for that. And the last thought that I wanted to share with you is that we had this opportunity to launch this startup company in Argentina, and this is because we had already worked a lot with RNA in plants, trying to figure out the roles of these long non-coding RNAs. But around 15 years ago, more or less, we knew already that plants can absorb RNA, and that in that way we could use this universal language to communicate with plants, so we can fry RNA on plants and plants would absorb it and breathe it. But when I was in France, it was difficult to find partners, strong partners, to develop nanotechnology to stabilize RNAs for crops. The main interest in Europe was, or the groups that were working with us, was pharma. So we just gave up during my postdoc. But once I came back to Argentina, I found that paying attention to what is going on in each country is very important, because Argentina is a country where there is an outstanding nanotechnology, but at the same time, it's a very important agricultural country. So there was a lot of nanotechnology developed for agriculture. And this is how, once we obtained a very important grant in Argentina to develop technologies based on RNAs for agriculture, we could interact with nanotechnologists from Argentina, and we found a way to stabilize RNA and to throw RNA to plants so that they can absorb it and read it. And this is how we started to develop RNA-based technologies to replace synthetic pesticides. And this happened in parallel with the pandemics. So RNA became like the central molecule in biotech, thanks to the vaccines. And what we are doing is something like that, to try to develop RNA solutions, based solutions to replace synthetic pesticides. That is something not only that agriculture is demanding, but also the society is demanding. We need to improve the way we produce food. And I found that Argentina, although there are a lot of problems for funding science, there is an opportunity there to launch a startup company, to have access to international grants or investment to grow, even though we are in a peripheral country. So I think that is important because we came up with an idea and we could undertake research to develop science and technology in my home country, always in collaboration with other countries, but we had the opportunity. I think networking is essential also for the startup, but I think that possibilities are there. I would suggest young scientists and enthusiasts to try to travel abroad, not only to central countries, but also to other countries, to know better what different people are doing, how different perspectives can understand the problem, and try to expand the networking across the globe because it's very, very helpful. And sometimes solutions or ideas may come up from places that we wouldn't expect. So, and also for that, my lab is always open for everybody who wants to join us. So just drop me a line if you're interested in our technology or science or whatever. I would be happy to collaborate. So thank you very much for the invitation, and I'm open for questions at the end of the session.
Thank you so much, Federico, for a very informative talk. Our second speaker is Dr. Sonia Negrao. She is an assistant professor at the University College Dublin and leads the crop stress interaction lab. Her research focuses on the improvement of crops tolerance to abiotic stress under changing climate conditions using plant-based imaging phenotyping and exploring underutilized genetic resources. She obtained her PhD at the Nova University Lisbon in Portugal, collaborating with the International Rice Research Institute in the Philippines. She then worked as a research scientist at the King Abdul University of Science and Technology in Saudi Arabia. Thank you so much, Sonia, for participating in the webinar.
Thank you, Villa. I'm going to share my screen. So I just got a very tiny presentation. I'm not going to bore you with too many details. I hope that you can all see it well. Yeah. Okay. So thank you so much for the lovely invitation. It's really a pleasure to be here. So I just thought about presenting a brief story of my life. So I was born in Portugal here, and I'm actually from Algarve. I'm not sure if you have heard about it. Lovely beaches. So this is actually my childhood beach. I lived a couple of 2 kilometers away from it, and I spent literally all of my time there. So this is just me. Very young girl, zero interest in plants. I actually at the time wanted to become a vet. I tried to go into vet school and I didn't have enough KO points. But I got passionate by genetics. And so because of genetics, I didn't get the KO points to study vet school, which thank God was a blessing, because now I know I had zero capacity to become a vet. And I fall in love with plants. So I did my bachelor's in agronomy and I majored in crop breeding. So you can see progressively me getting older throughout the photos. So this was done in Lisbon. And then in my the equivalent to nowadays research masters, I had an opportunity to study in a very well-known institute in Portugal, which is called ITQB and belongs to the University Nova de Lisboa. So I did then my PhD in collaboration with the International Rice Research Institute in the Philippines. And I spent several months living in the Philippines. It was there that I made my rice crosses. I worked a lot in the field in Portugal and I collaborated with several people. I also lived, as I mentioned, for several months in the Philippines and was able to travel just a tiny bit in the Philippines. And Philippines for me, so I have a deep passion about the country and the people, and it became like a sort of a second home for me. So then I continue my research in Portugal, and I continued in collaboration with the International Rice Research Institute in the Philippines. So this is a photo from there. This is also photos from the Philippines. This is a photo of me in the field in the middle of Portugal, where we had a rice salinity trial, and you can see that rice was scoping it very poorly. And then things happened. A cow jumped the fence and ate my essay. So I lost all of that year of work. So that's one of my advice. Do not give up. Everything can happen. And everything has happened to me because I work a lot in the field. So never give up. Then I had the opportunity to move into Saudi Arabia, where I lived there for 5 years. I joined Professor Mark Tester lab, the salt lab, and as you can see, we had a very diverse cohort of people in the lab. My husband and I, we went there, the two of us, and I want to mention that if you want to move, one of the most important things that have to happen is the support of your core family. And when I say core family, it's not your friends, it's the really core family. So both my mom and my sister, and especially my husband, because my husband gave up on his job so that we could move to Saudi Arabia. And he had a permanent position. So we risk a lot, and sometimes we have to risk it because the higher the risk, the higher the gain. So we stayed there for 5 years, and in between, we became parents. So I'm a mother, proud mother of 2 twin boys. And you can see me here working. I work in different crops. I continue to work with rice. Rice was always my passion, but I started working with Solanum pimpinellifolium, which is a wild relative of tomato. You can see me here in the field. And you could see me here as well. This was a trial that we had in Dubai. So this is Dubai field, and we are working with barley here. So then when I moved, so I stayed there for 5 years, and at the end of my final year there, it was time for me to move on. I was mature. I was ready to start my own lab, so I applied for several positions. I got a couple of interviews. I learned in the process of interviews as well. So sometimes you go to your first interview and it doesn't go well. And it's just because you didn't know how to answer well and you were not skilled enough. And these are the types of things that you need also to train yourself. So I trained myself. I learned from my mistakes. And then in 2018, I started working in Ireland at University College Dublin now, where I am based. So I developed my lab, the Crop Stress Interaction Lab at UCD, where we do lots of interdisciplinary research. So we used imaging sensors in the field, so we fly drones. We also have imaging sensors in controlled conditions. I established a strong network, and again, networking are super important. Opposite to what Federico was telling, when I moved from Saudi is a desert. I moved to Ireland, where it rains all the time. And I was working with rice. So suddenly it didn't make sense at all. There's no rice in Ireland. There's no salinity didn't make sense at all because it rains so much, unless I would go to more the ecological side of it, which was not my passion. I like to work with crops because my background is breeding and agronomy. So I had to change entirely. One of the things that I want to mention as well is the support of the PIs. So Professor Mark Tester was awesome. I told him about this opportunity. At the day that I had the job interview, I was supposed to be giving a talk at PAG, the Plant and Animal Genomics in San Diego. So he said, you go to the job interview. I will give the talk in San Diego. So I went to the job interview. He gave the talk on my behalf. And when I arrived, in San Diego, everybody knew that I went for a job interview. So it was it was fun. Basically, it was important to have also the support of the PI. So the PI at the time, because I started working with Barley with him, I was able to also start knowing people in Barley. So I established collaborations with them when I started my lab. I wrote to people that Mark had introduced me to, had discussions with Mark, what would be the interesting part to look at. And I told him, I think I want to look at water logging because I'm in Ireland and it makes sense, water logging, because it's constantly raining. And he said, that's an amazing idea. I think there's awesome research being done at very fundamental level, awesome research being done at the breeding side of it. But then the other part, it's a little bit understudied. So I think it's a nice idea for you to look at. And that's how I started my lab. I was fortunate enough to apply for big funding in Ireland, given by the President of Ireland. So I went to a ceremony where I received an award from the President of Ireland, Michael Egan's. It was an award that allowed me also to expand collaborations. So these interdisciplinary projects, I have collaborators in UK at the James Hutton Institute and now the newly established International Barley Hub. I have collaborators in France, Université Picardie Jules Verne in Amiens, which has been fantastic as well. And I have established collaborations as well because I'm in UCD. And there's a massive school of computer science, and IT is very big in Ireland, as you might know. All the big social media platforms have their European bases in Dublin. So there's loads of IT and software development. So I established also collaborations with Professor Eleni Mancina, School of Computer Science. My group fundamentally works in these areas, but then I still continue to like collaborating and testing the limits. So I'm now, my new chapter is that I established cost action that I'm leading. And at the moment we have 48 countries participating on it. And this is one of the brilliant things of Europe. When I was away and I was in Saudi Arabia, I was not, I was a little bit out of the game. And it's really fantastic. So in this project, I joined forces with archaeology. Again, the networking in UCD opened the doors to it. And there's lots of interdisciplinary research as well. So with nutrition and policy as well. So I never thought that I would start collaborating so much with arts and humanity. But now it's actually, it's very interesting, and it opens lots of doors in terms of research because there are lots of things that we never thought. And I think that new ways of thinking, new brains really help a lot, and diversity is fundamental. And I think I've finished my presentation. I'm just going to stop sharing my screen if I can. Yeah. And yeah, so basically, I think I worked in all the continents. So although for shorter periods of time, I was also based in Australia for like 3 weeks. I was based in Senegal for a week. I was in the Professor Michael Puruganan's lab for like 2 or 3 weeks as well in New York University. So although I have, what I presented to you here was the places that I stayed longer for more than a couple of months. But I think that, and if you asked me a couple of years ago about it, I would say I would never leave Portugal, not in a million years. I'm very attached to my family. I'm very Latin, very conservative. But in fact, it's that if I would have stayed, I would never have progressed. And I think that I like trying new things and push the limits. And I I felt that I needed to leave. And again, the support from the core family was fundamental. And my husband was the first one to say, if you stay here in Portugal, you don't have many opportunities, we need to go. And I had the job invitation from Professor Mark Tester. He was establishing his lab from scratch and invited me to become his deputy. And it was an opportunity that was asked 1 week before I got married. So the last week, my husband and I at the time spent a whole week not talking about wedding plans, but should we go to Saudi Arabia? Yes or no? So yeah, so that's my message. Never close the doors. If you go to a conference, treat that conference as a job interview, because that's what happened to me. I was totally unaware of it. I went to a conference, I did my oral presentation. Professor Mark Tester was sitting in the audience, and I was afraid because he was the salinity person. I kept on seeing him nodding, and in the end, he asked me a couple of questions. I answered. He said, "Oh, it was a great presentation. I'd love to catch up with you at a coffee break." And we start talking. And basically, he in the end of the conversation, he said, like, "I would love if you would join my lab." So that's it. The opportunities can come, you never know when. So do not close the doors to those opportunities. Yeah, that's me. Thank you. I'm welcome for all the other questions.
Thank you so much, Sonia. That was a beautiful telling of your journey so far. I'd like to introduce now Dr. Felipe Kliemann, and he is currently an associate professor in the Department of Botany at the Federal University of Rio Grande do Sul, where I did my PhD. And the reason I invited Felipe is not only because of the quality of his research, which revolves mainly around ion mechanisms of ion homeostasis in plants, but also because of the approach he brings to his research, which I see as multifaceted and very collaborative. And I suspect that it was that view of science was at least partially developed in his journey through different countries and interacting with different people. So thank you so much for accepting my invitation, Felipe, and the floor is yours.
Thank you very much, John. I'm going to start sharing my screen here, and also thank you all for being here and also for the invitation. I'm very honored to be here with all of you discussing a little bit about plant science and about moving around and how can we improve the way we do science by moving. So I made this attempt to be a funny title that, you know, I really think that you should move if you have the opportunity. And I think Federico and Sonia already stressed that point. So before I start, I made a presentation about like my career and why I might be talking about moving in a good sense for you all. So the first question is like, who am I? Where do I come from? How did I started working with what I work? So I did my bachelor's degree in the same university where I'm based right now in biological sciences. And at that time, just like Sonia, I had absolutely no interest in plants. I actually skipped most of the classes on plants. I was working in 3 different labs at different times. I worked with fruit flies and also with parasites and also with neuroscience using rats as a model. And I had no interest in plants. But during one time that I was doing one class, it was an advanced class where we were discussing about the golden rice. So most of you may know about golden rice. Is this biofortified version of rice where they improved the concentration of vitamin A and of beta-carotene in rice. And there was one research which became my advisor during my master's degree in the same university. She was planning to discover some genes that could increase the concentration of iron in rice as well. So I was very fascinated with the impact that could have. We know that billions of people today have iron deficiency, anemia, in their diets. So I got very, very excited about that. And I decided to join the lab for my master's. But at that time, I was all the time joking that I was interested in the result, not in the plants. So plants were not very fun to me. I actually like molecular biology and genetics much more than I like plants. But due to that, I was still very young. I finished my master's and I was not sure that I should go to doing a PhD. So I got a job as a temporary professor of biochemistry in my hometown, which is in the center of the southmost state of Brazil. I'll show you the map in just a few slides. And I also worked for a company as a product and sales manager. That was a horrible experience. I discovered that I really belong in academia, but it was good to know. And then I came back for my PhD in the same lab with my advisor. So I was working here in the corner, in the southmost corner of Brazil with Professor Jeanette Fett. She was an amazing advisor to me, gave me a lot of opportunities and advice. And one of the best things that ever happened to me is that she got an NSF grant to send one student for 3 months to David Salt lab at Purdue University. He was the guy that started the idea of the ionome in plants a few years before, and also 3 months in Mary Lou Guerino's lab at the Dartmouth College. So Jeanette sent me for these 2 trips. I spent 3 months in each lab, and then I put that back to back with another year in Mary Lou's lab using a Brazilian fellowship that I was awarded. So during this time, Mary Lou was an excellent advisor. David was an excellent advisor as well. Mary Lou sent me to multiple trips to the Brookhaven lab to do some synchrotron research and also to Stanford. And with that, we got 2 papers out of my PhD thesis. I also did a lot of collaboration with her group, especially with Tracy Punchon, which is a dear friend of mine. We still collaborate. All the 3 of us, we all collaborate to this very day. And I want to stress how important that is. Mary Lou, in 15 months, she allowed me to go to 5 different conferences. She actually paid for me to go to those. So I was exposed to a lot of people in a lot of different contexts. In 2 of them, I could present my own work. I already mentioned that I was in 5 different synchrotron trips. So I was doing experiments in institutions that I've never thought I could go at that time. She gave me a lot of freedom in the lab. I could do essentially everything I wanted, and also a lot of that advice. And I want to stress that they all still give me support on my research. We still collaborate, we still publish together and exchange ideas and everything. So then I went back to Brazil. I finished my PhD and I started as a postdoc in the same lab that I was with Dr. Jeanette Fett for 2 years. But of course, I could not stay put for a very long time. From that time, we got some grants with David Salt. He had recently moved to Aberdeen University in Scotland. So I got the opportunity to go to Scotland, experience a very, very different context for doing research. It was an amazing opportunity. I met a lot of good people there. And then I moved from Brazil to Dartmouth College back again as a postdoc. I had a 2-year fellowship, but when I arrived in the US, I actually got a job back in Brazil. So this is in another federal university, is in the center of our state. And I started my own group there in 2015. That was a very good moment. I recently, well, not that recently anymore, but at the beginning of the pandemic, I moved to my alma mater in the Federal University of Rio Grande do Sul, and I'm actually now in the same lab where I did my PhD. What happened during this time is that I had my daughter in 2016. This is Maya, my beloved daughter, and because of that, I saw all the impact that the arrival of my daughter in our family had on my wife's career. And I joined. I was fortunate enough to be invited to the Parenting Science movement, which is having a lot of impact in Brazilian science, talking about how science impacts in an unbalanced way the mother's career. And I've been trying to be together with the movement, but this is mostly done by the people that move parenting science, especially our head, which is Fernanda Stanisławski. And I hope you can access the website and learn a bit about parenting science if you have the time. So, sorry, 3 things happened during this time that were amazing for my career. One of them was I got a grant for early career scientists. It was a very competitive one from the Serra Pilheira Institute in 2018. A couple of years later, I was elected for the Brazilian Academy of Science. This is a under 40 election that they have, so this is a temporary time that you spend with the academy. But it was very good for my career to get that. I'm very thankful for Marcia Marges, which indicated me for that. And in 2022, I got a Fulbright scholarship to go to Pam Ronald's lab. I saw her picture here in the beginning of our webinar, and I want to point out that both this one grant and the Fulbright one are in collaboration with Pam Ronald. Pam received me in her lab in 2023. I spent 4 months in her lab. It was an amazing person to get in touch with. I always want to remind people that this started with me seeing the grant open, and I've never met Pam before. We never crossed paths, and then I just sent an email to her. Here, I have an idea. I would like to work with you. And in 5 minutes, she replied back and said, yes, of course, that's a great idea. And then on, now I have one previous PhD student that is a researcher in her lab. So I'm always thankful for that. So she's an amazing person. Anyway, where I am right now, this is the southmost state of Brazil. This is where my lab is based. I moved from Santa Maria, which is in the center of the state, to Porto Alegre, which is the capital city. This is our beautiful city. I think John might be a little feeling a little sorry that he's not here. It's been a while that I don't see you here. This is our university. We have some old buildings, of course, for European standards that those are not very old, but I really like our university. You should come here and visit at some point. And I want to point out what we're doing right now in the lab. So this is my team. I'm very thankful for all of them. This is my our new postdoc researcher, Dr. Ortolan. She joined the lab a couple months before. We have now 5 PhD candidates that are working in the lab in different stages, and also 1 undergrad that is working with our team. And I don't want to go too much into the research that we do, but our main focus is trying to understand what the ionome is and what how the ionome is regulated. So for those of you that are not familiar with the idea, the ionome is essentially the inorganic composition of an organism. And of course, we work with plants, and we try to take this, our research in a very integrated way. We use a lot of different tools to understand the ionome and how the elements and how the molecular mechanisms that regulate the ionome interact. So this is the image that I like to keep on the back of my head that we're trying to understand. We want to know which element interacts with the other and how they balance each other inside a plant's body. So we're interested in a lot of stuff. We use rice as our main model. As you may not know, Brazil is the main producer of rice outside Asia, and our state produces almost 70% of all rice in Brazil. So we're very interested in this crop. We also, of course, work with Arabidopsis and a lot of different tools. We have some wild relative work that's been going on here, but I don't want to spend too much time here. So to finish, what I think is our main points, we can discuss more later, but what I think is good about Brazil and why I think it's good for me to be here. First is job security. Brazil has, if you get a federal job, this is a very secure job. So you essentially you are tenured. So this is very good. You can you can do some slow science if you want. There's no problem on that. Of course, grants are not going to be coming your way as fast as you want, but it's a good point. The second thing that I like is that competition sometimes is in the middle range. You are valued if you're publishing this sometimes incomplete stories that you cannot advance because you either don't have the people that are interested in that, or you don't have the money for it. And I think sometimes this is undervalued in more competitive environments. So I really like that. I don't mean to do bad science, but I think sometimes it's really important for you to publish your paper and get your students to actually learn how to publish and show that they can finish stuff. And of course, the impact that you have on students is very palpable. I can see that, you know, I will be just another researcher if I was in the US or in Europe, but I think in Brazil, you were more needed. We need a lot of good people that are passionate about science here. So I can see that I can actually take some students that are starting their PhD, given an opportunity to go to the US, and they actually take it and they fly from that. So I think this is very good for working here. What I don't like is that we have grant insecurity. It's very hard to have a steady stream of money in one lab in Brazil. The institutional support is very low, so sometimes we actually have to fix things ourselves. And we're far from centers where science is happening. So even for conferences, it's always very expensive for us to go. And what I miss about being abroad is, of course, access to platforms, equipment, grants, and also people, like the people that are actually doing stuff. You were just, one corridor away, one knock on the door away, and this is very good. And of course, the institutional support that is more common in more developed centers. And what I learned from that during all the time that I was doing research, and I think it's like just 3 words that I would actually stress for you all, is that you should move. Just go find different. Not everything is going to be good. Sometimes you're not going to like it, but you're going to learn from that. I also think it's very, very important for you to speak. If you have an opportunity to present your work and an oral presentation or anything else, just take it. I took a lot of the opportunities on that, and I became less and less shy to do it. And I think you should do it and try to connect with people. Just try to be close to the people you admire. Try to talk to them. Usually 99% of people in science, they're very open for people that want to talk about the research. And so just send emails, get them on conferences and talk to them. So with that, I'll finish. There's a lot of people to thank for all the support that I've always had. And I want to point out that you can find me anywhere, and we're always looking for interested people in our research. So thank you very much.
Thank you, Felipe. It's really nice hearing from you, and also thank you to the other speakers who in the meantime have been answering lots of your questions on the Q&A. We still have some minutes, not many, but we still have a brief time for questions. So I guess we can have brief answers from well, actually I'll point this to Federico if you don't mind. Could you share with us maybe a particular cultural experience that you had that in another country, for example, where you could tell that the research culture was different between, for example, maybe Argentina and France? Is that something you came across?
Well, actually, in terms of culture, France and Argentina are very alike. But what I found is that the French Institute was much more diverse than my former lab in Argentina. And I had to share the lab and the institute with people from Asia, other countries from Europe, or American, or even African countries. And that was very, very interesting. I think that we learn how to communicate with people, even try to overcome barriers. I think we learn how to be more patient and how to understand the other's perspective on things. And then that is not only about the subject that we are studying, it's also about, I would say, everything. I mean, the way we book an equipment to use. I mean, there are rules in other cultures that perhaps are not the same rules that we have. For example, Argentinians, we are very impunctual. We arrive late everywhere. But that is something we have to deal with when we are in another country. And sometimes there are other things that may sound weird from other countries, and it's very interesting and sometimes even funny, but it's very, very nice to learn about other countries' culture. And I think that was very important. It opened my mind a lot to share with people from different backgrounds, the lab, and the opportunity to discuss and to learn about their history, their culture, their geography, and to find that different countries face different problems in terms of plant sciences and agriculture. So that I think it's very, very important. And this is why I try to keep my lab and our institute very international, the most as we can, because it's very, very rich to share experiences with other people.
Thank you for your answer, Federico. I will now throw it to Vilo if you want to ask a question, maybe to Sonia. I don't know if Vilo is with us, or maybe her connection is not so good. Otherwise, I can ask the next question, which so Sonia, we got a question about that touches on the topic of language barriers. Is that something that you feel like was a challenge for you, or maybe did you see it be a challenge to other people around you?
Yeah, it's a challenge it was a challenge for me in the beginning because I'm a Portuguese native speaker. And the first time I went to the Philippines, I struggled a little bit in the sense that I was not proficient. But I just tried and improved as much as possible. And nowadays I see that with lots of students. UCD is very international. We have students from China, India, several countries in Africa, of course, Irish students. And I can see several students and even people in my lab. Just very few of them are native speakers, only the Irish are native speakers. So language barrier, yes. But one of the things that I recommend to students to do, it's watch Netflix shows, Disney shows, whatever, with English subtitles. Watch it in English with English subtitles. That really helps a lot for you to improve the English, just by hearing the sound and seeing what is written down so that you don't get lost. Yeah, but for example, in the Philippines, there were several times that it was quite challenging. And I remember that I had a collaborator from Japan, and my supervisors, I had a meeting and my supervisor said, "Oh, I'm going with you to the meeting." And I was like, "Why are you like, I can handle this?" And he said, no, it's just because the language could be a little bit harder, so it's much easier if I'm if I'm there, just because to translate a little bit. So yeah, I have encountered, yes.
Yeah, thank you, Sonia. I'm really sorry to get this short. We're running a little bit over on time, but by no means this means that the discussion has to end. You have access to the bios of all our speakers, and Plantae is all about this, about open collaboration and discussion. So please do keep the topic going. Thank you all again for attending, and thank you the speakers for accepting our invitations. Thank you to my Plantae fellows and Plantae fellows, and especially Ileana, who's gone above and beyond for this. And now all that remains is for me to throw it to Jason to see us off.
Great. Yes, thank you, everyone. Thank you to our speaker. Thank you to our moderator. And of course, thank you to all of you who attended this webinar. It's been a very insightful webinar session, and I hope that you will tune into the next one. I know we have questions about be shared with you after the webinar. You'll get an email, and so just kind of follow that instruction and how to get the certificate. And with that said, I hope everyone have a great weekend, and we hope we see you until the next one. Again, thank you to our speaker, moderators, and to all of you. Till next time, have a good one, everyone. Bye.